Last time, we pulled a thread from onion juice. I bet everybody here knows what that thread was. It was DNA, wasn't it? And there it is on the left, and on the right's the thread of my DNA. They're almost exactly the same, those two threads, but the subtle differences between them mean that one makes an onion and the other makes a human being. The DNA in this baby's cells, this is Max, by the way, the DNA in Max's cells made him grow and become a child, and that's what we're going to explore today. And the DNA in Max's cells is buried deep in the heart of them, in the nucleus. We need 2 volunteers here who are going to come and pull open the nucleus in a moment, but wait for it. Inside the nucleus are the chromosomes to make a human being. So if you grab hold of it, and I'll give you a count to open it, you grab hold here and get ready to pull when I say. So it's 3, 2, 1, and open. And there are the 46 chromosomes of human DNA. So now if we pull this up a little out of the way, that's it. And then we need to sort these out. They come in pairs because half of them come from the mother and half from the father. And we have some people who are going to come down and help us sort them out, I think. And the idea is that there's 23 of you, including you two, you're part of the gang. And you're going to sort them out, pair them up by color, and see how you do. That's right, keep going. Thing is to grab one and then try and find its partner. Of course, you might grab 2 pairs. You'll just have to be clever about that. And here on the screen, you can see a picture at very high resolution, scanning electron microscopy of real chromosomes as they are, flying across the landscape of those real chromosomes. How are you doing? Have you got those sorted yet? Right. Has anybody not got a pair? Now you see there's one person who has an unpaired set of chromosomes. Does anybody know why that is? Max is a boy. This is the X and the Y chromosome. A girl has 2 of these X chromosomes, but a boy has only 1. And in addition, he has this fiddling little Y chromosome. And worse than that is just 1 gene on there, basically, that makes him a boy. So a boy is a kind of a slightly modified girl. The girls may feel that actually it's not a very successful modification either. Now what I want you to do is to get in a big horseshoe and face outwards, and I want you all to throw your chromosomes up to the very back row of the lecture theatre. That's it. Well done. And now the volunteers go back to your seat. And meanwhile, I want you to pull out the DNA that's in the chromosomes. And I want it everywhere. There's a lot of DNA. I want it everywhere and coming down towards the front. Every cell in the human body has 2 meters of DNA. Literally 2 meters. You're scaled up a bit here with this stuff. And in all the cells of the human body, there is enough DNA, if it was laid end to end, to go 500 times to the sun and back. And here we see on the screen, again, the reality of DNA spilling out of a human chromosome. There's the tattered remains. It's been treated with detergent. And the DNA, see the lines of DNA spilling out, and you're creating that with your strands of DNA pulling out from those chromosomes. But first, we have to go back and think about what DNA really is like. And it's like this. It's a spiral with 2 strands, and if we straighten it out, it's easier to see. There's 2 backbones, one going one way and one the other, and in between there are these pairs of bases. And they come in 4 sorts. you have thymine, you have cytosine, you have guanine, and adenine. And they can form pairs. In fact, they always form pairs when they're in the middle of the DNA, and the pairs are like that. And these white bits are where the backbones go up. And what was discovered, the key thing about the structure of DNA when it was discovered nearly 50 years ago, is that the two sorts of base pairs fit just on top of each other. And so when you make DNA, the base pairs fit and make this nice regular spiral. We don't say those long words all the time. We call them T, A, C, and G for shortness. Now, the clever bit about these bases is that this is the information to make life. We call it the sequence. And by that we mean the order and the sort of bases along one of the strands. And then because of the base pairing in the middle, the other strand is defined always by the first strand. And so we just think of the sequence as being the order of the bases along one of the strands. We have a model here which shows how it more really is, which is if I was an atom looking at a molecule of DNA and I could sort of feel at that atomic resolution what it was like, then this is what I would see. It's more solidly filled. You can't see the base pairs in there, so that's why we have the models so that we can see more clearly. Now, what I want to do is to think about the sort of information that's being stored there. And if I could get you to hold the end of that, if you put a hand top and bottom so we can see it, and stretch it out so everybody can see. And perhaps Max can come back for another quick. How are you doing, Max? You all right? Can you bear it? There he is. So what we're looking at is how Max was made. Now, one way of making Max would be to have a blueprint. And that's what we've got here. There's architectural drawings of Max. You see, side, head, elevation, all that. That would be one way. In fact, at one time, people thought that was how it was done. They thought they could see a little man inside the sperm, and that this would grow and become a human. The other way is this. And that's what we've been looking at with the ladder. That's DNA sequence scrolling past there on that board. And we're going to have a look at some other things, some other ways of coding, so that you see, because this way, the blueprint way, we can say is analog. Analog means that the way of coding it is rather like the thing itself. Digital, on the other hand, is not at all. It's a code, and it's only by some prearranged understanding that you can see what the code is meant to be. So I think I'd better put Max back, otherwise I can't show you the stuff. But just bear in mind that it's Max that we're making out of all of this. Now you see, here's some other analog things. Look at this chap. Is he analog or digital? Shout out. Analog, exactly. He looks like, well, I don't know if he looks like, but he's supposed to be looking like the person that he represents. How about this? Analog. What about this? Is this analog or digital? Digital, exactly. And this? Digital. Not quite so sure about that, were you? But it's the English language, and it is digital. It's just characters. They're meaningless. They don't look in the least like what you're trying to represent, but we understand, we have dictionaries to understand what we mean by them. And with this, is this analog or digital? Digital. Digital, it's a computer. It has its own code, which is binary. It has 2 symbols, unlike DNA, which has 4. But it very much is the same. And finally, oh, these 2 are nice. This one is digital. Analog, yes. Now, there's only a few people perhaps know that. This is an older sort of gramophone record. We mostly have CDs, which are digital, because they have the same computer language. And there's the analog gramophone record. And the wavy grooves on this represent and make the sound, when it's played back, in exactly the same way as the sound waves coming to the ear. So when the sound is recorded, you get waves like the waves in the ear, in the air, and when it's played back, then the waves go out again. So there's the analog, and then the digital where the sound is analysed in terms of numbers, which are then printed on in a binary code to the CD. We'll take these off again, I think. Now, when it was found that this was a digital code, it was absolutely astonishing, because life, after all, is a rather sort of general, squishy thing, very complex, and people felt, until DNA was really firmly identified as being the source, they felt that it must be coded in some much more complex way, the information to make it. But this actually is really digital. It's exactly analogous to the sort of software that we put in a computer. Does anybody know how to play Tomb Raiders? Come on then, who's there? Come on, you. That's good. So you can play that and show us how it's done. Now, in order to play Tomb Raiders, you have to put in software to the computer. The software has a code, just like our DNA. But the clever and paradoxical thing about life is that the DNA does much more than that. It actually makes the computer itself. Now, you'd be pretty surprised if you put in some computer software and you got a new computer. You know you have to go to a shop to buy a new one. But we're not like that. We're actually built our hardware, ourselves, our body, our structure is built by the code, as well as the software part which runs us. What we're going to do now is to go on to the most important thing. I think I'll take your blueprint back, shall I, out of your way, so you can see what's going on. The most important and central thing about DNA, apart from the storage of information, is that it has the ability to copy itself. And I think we've got a pool of nucleotides here who are going to come down and stand in the middle. Is that right? You ready? And we also have Sticky here, who, as well as being our gallant floor manager, is now going to act as DNA polymerase, and you'll see how it works. And now here, if you stand up, we've got a DNA helix, a DNA double strand waiting to be replicated. Hang on there for a minute. Now remember, when you come down, you're going to hold on to the person in front of you. Even when the strands separate, you're going to hold on to that shoulder in front of you. We want you to stay together, and we won't take it too fast. So what's going to happen is Sticky is going to take control as the polymerase, and he is going to beat time as you replicate. And now as the strands separate around Sticky, because of the base pairing, they can each pick up, hold onto the shoulder in front, keep those strands together. They can each pick up, see how they're each picking up the complementary base just according to the pattern on there. That's right. Keep going. That's right. Good, 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 good. And there we are. We've replicated our DNA. And as they march out of the door, you see life in action. That is the process. Well done. You did a perfect job. And thank you, Sticky. You are a perfect polymerase. So the DNA, this is how life soldiers on. The DNA is being replicated, but remember, it's not just software, it's hardware. So the new DNA goes into new cells. The cell can divide and start to make another part of the organism. And so the DNA, the replication of DNA, is the heart of life. And this is how the code works to build us. And in a moment, we'll see how we go further from that.
From now on, we'll celebrate New Year with dignity. We'll drink copiously, but only water. We'll only ever eat sensible food. We'll go to bed earlier. We'll treat body as temple. We'll go to gym at least once a week. Second thoughts, make that twice. Rejuvenation begins. Step 1, use new Boots Detox. Step 2, apply Natural Collection face mask. Step 3, use Boots Alternatives. Feel like a new woman, plus have created virtual health farm in own home, without any scary procedures.
Good news! Not only have Johnson's baby wipes triumphed in kissometer testing, yes, they won a Mother and Baby Gold Award too.
So, thanks, Laura. You're finishing our tower for us. We'll find out in a moment what this is for, but first of all, I'm going to ask you if you'd like to pull this string rather hard. I think you'll have to get up to do that if you come and stand here. Are you ready? Give it a yank. Pull hard. Way! Well done. Okay. Well, take that away, and you'll see the point of it in a little while. But meanwhile, we've got all this DNA everywhere, haven't we? It seems to have sort of got assimilated into the audience. I hope you're not eating it. How many people ate DNA for breakfast? Well, the answer is you all did. If you ate anything, it would have had DNA in. But anyway, don't eat this. It's not so good for you, this sort. So how do we deal with this DNA stuff? We've got an enormous amount of it. It's very seriously big. The number of letters in the whole of our code occupying the 1 meter that makes a single copy, because remember there's 2 copies, one for your mother, one for your father. Each copy has 3,000 million of these letters in. That's a seriously big thing to deal with. And that's why we haven't sort of got to doing it until very recently. And indeed, without a number of tricks, it will be quite impossible. Because we've got all this mess. How do we deal with mess? How do we get hold of a piece of this huge, long, tangled, unique stuff? Well, I'll show you. Give me a bit of this stuff. Let's tidy up a bit here. Let's take some. We chop it up into small pieces. That doesn't seem a very good way to begin, does it? But that's exactly how we start. We can chop it up in a couple of ways. One is to use special proteins called enzymes that know to cut at particular places along the sequence, and with those we can make quite long pieces, which can be useful. And we can also use this gadget, which buzzes the DNA so that the molecules shake around and stretch, and they physically break in the middle, and with that you can get much smaller pieces. We use both those methods, but in both cases we still haven't got anywhere. All I've done is to chop it up. We've still got this mess. What we have to do, and this was the trick that allowed people to get going with DNA, is to hook it in to another special kind of DNA. Here we've got it coloured red, and this DNA is called vector. And what it's able to do is to carry the silver DNA, the human DNA, into bacteria and to allow it to replicate as the bacteria do. What's happening here is the bacterial DNA is dividing, the cells are dividing, the bacteria are multiplying, and this is multiplying with it, many copies per cell. And now, suddenly, the problem is much easier, because instead of having single isolated bits all different from this huge long sequence, we've cloned, we've replicated one particular bit many times, and we can go on and replicate it as many times as we like just by growing those bacteria more and more and more. Now, this has allowed us to set forth on this Human Genome Project to actually collect all the bases. And when we do it, actually, in order to make the task manageable, we use both methods. We first cut it down into those large pieces, and then we take each of those large pieces and cut it down with this gadget into many small ones. That way we don't get muddled between things that are far apart. We're dealing with a section at a time. And it's worked very well and has allowed the full code to be collected, we'll see in a moment. But how do we go about doing it next? Well, the next thing is to put this into bacteria. We use an electric shock to do that. And then we spread this mixture of things out on a plate. Now remember, at this point, I haven't got a single clone. What I've got is a mixture of these pieces, because each of them has got linked into one of these red bits. So to sort them out, what we do is to take the bacteria and spread them very thinly, and then allow them to grow. And maybe you can see that there are spots on this plate. And if I put it under the microscope now in a moment, you can see very clearly, if we get the feed from this microscope, please, you can see the colonies of bacteria, and there they are, growing nicely on the plate, all under control. And so that's how we clone things. The act of spreading those individual bacterial cells out and then allowing them to grow, each of those round circles, is a great mass, just like you saw the one dividing in the time-lapse film in the beginning. So now we can have a look and see where we go further with that. These machines are picking the colonies. Here is a device with many needles working at the Sanger Center. It's imaged the picture of the plate. You can see the colonies, and each pin is stabbing down into a single one of those. And then in a moment, it will move across and inject it into a growth box, which then is shaken, allows them cells to grow up much more. The DNA can be extracted from them all and is carried over to another robot where the sequencing reaction is done. And what that means is that it's made into a form where we can usefully put it into this machine. The bases have been labeled and the strands have been copied starting a particular starting point, so we can actually measure how far each base is from a fixed starting point. And that's what you can see coming out on that screen there. And up above, you're seeing a webcam feed from the Sanger Center where the staff have specially gathered to greet us as we talk about what they're up to. Normally, you won't see anybody here because there are just these machines with one or two people walking around, but they're all there saying hi. So that's coming to you live over the web. But meanwhile, from this machine, which is actually running DNA as we speak, you can see on the screen here, and I hope we're picking that up if I stand over here so you can. I hope we can pick that up and see the peaks, the colored peaks marching across the screen. And those colored peaks correspond directly to reading out a sequence of bases, just like this. And they're color-coded so that the computers can pick up which base is where and interpret it for us more or less, although we have to do a little bit of tidying up afterwards. Then as a result, we can get this human sequence really scrolling here, but there's a step to go yet. All of this process, the cloning and the sequencing, only allow us to read out a few hundred bases at one time. And we've got 3,000 million to do. We've got many thousand even in one of these large pieces I mentioned. So in order to see how we go about that, I need to have a couple of assemblers. I need a couple of volunteers to come and do some assembly. So would you like to come? Okay. And where are we? Yes, you. Would you like to come? Yes, in the purple sleeves. Now, I have some things here for you to assemble. Quite heavy, actually. These are pieces of sequence. Now, I told you there are a few hundred bases, so you haven't got quite that much to do. But they're a sort of a jigsaw puzzle. And what you're going to do is to look for the overlaps between those. There's a unique way of putting these together, just with 3 bases overlapped on each, okay? So can you take a look around and see if you can find those overlaps? When you get them right, there's some pegs which will fit there so that you can, like that, so that you can fit them together, all right? So see how you go. The whole idea is to take a lot of random pieces. Remember, we don't know where the initial pieces are when we do the cloning. We clone them first, pick them randomly, then we find out where they belong. It sounds mad. People often say, well, why don't you just start at one end and go along to the other? The answer is we can't. We have no way of doing other than this random process. And so you have to have an assembly process to put them together. How are you doing? You found any matches yet? See if you can find some as well. Have an extra volunteer here. That's good. Excellent. It's quite hard, isn't it? That's why we have a lot of computers in the sequencing business. They have to search all the time for these matches. And in fact, it's quite a sophisticated business that's driving computation to be able to find matches in a very large number of possibilities. It takes a lot of computer time to do that. Now this protein, this sequence I mean, that they're putting together makes a protein called keratin. it's the protein which is very abundant in hair, or at least it's a part of it. It's not the whole sequence to make keratin, but it's a part of it. So it really is a piece of human sequence, this, that they're putting together. How are you doing here? Time we got moving along. Look, that one goes here. That's it. You've only got one more join to make, I think, now, so it must be obvious. There we are. They're nearly there. They're making the last join. It's very exciting for sequence people when the final join is made. We say we're contiguous. Finish the DNA. Have you done it? It's there? Good. Well done. Now, so of course in real life things are much larger and they're not on bits of wood, and you can imagine how much we really do need computers for this. So here's a real life assembly that you can see here. These are the sequences, now hundreds of bases long, as they really are, the individual sequence reads that have been got out, and you can see the assembly with all of the letters nicely lined up, just like our volunteers were doing now. And you can see too that every now and again something goes wrong. A base had to be missed out here because there weren't quite enough bases. And if we look down in the traces below, because what happens is that we can display the raw data coming out of the machine, those peaks that are emerging are preserved, and we can look if necessary and see. And here we can see down here what's gone wrong. There should be a T followed by 1, 2, 3 Cs, and for some reason, because that was a little bit small, it didn't get called properly. So there was a gap in that sequence. It used to be that people had to edit all these things by hand, but more and more computers can take over that task, and the computer that assembled this would certainly have recognized that that base should have been there, and it would have sorted it out. But we still have to have people we call finishers who carefully improve the sequence and fill the most difficult bits and do additional sequencing reactions where necessary. And the result of all this is that a year ago we were able to release the draft sequence of the human genome. We didn't actually release it all at once because in the Human Genome Project we keep releasing the data all the time. The DNA sequence belongs to all of us. We share our DNA sequence with one another, and so it's obvious that it should be owned by all of us. And so what we do is to put it out on the internet for everybody to use. And here we see scrolling across the sequence from chromosome 22 of that sequence of last year, because we were able at that time to write it on a CD-ROM, and it's a rather remarkable thing, isn't it, that it's possible to write the instructions to make a human being on an ordinary CD-ROM, which you can put into your PC. It's running here now and showing you this program. If you look for a while, you will see red ones going by. And you can see red ones aren't very frequent. And the reason for that is that red stands for the parts of the genome that code for protein. These are the key central parts of genes, and they only occupy about 1.5% of the whole genome. And so there's an awful lot of other parts of the genome which is either totally unimportant or is not used very often. The next step, or the current step for the Human Genome Project, is to complete that. This is the finishing I've referred to. We did this quick draft, which is useful but imperfect. It has gaps and needs improvement, but it'll all be finished and put together properly by 2003, which is rather nice because that's the 50th anniversary of the discovery of the structure of DNA. Here's an animation that shows the process of readout. The DNA is there, and approaching it now is an RNA polymerase molecule which is going to open up the DNA strand, and it's going to make a copy, just like you did a little while ago with the replication, only this time the copying is not into DNA and it's only on one strand. These are making RNA. It's a very closely related molecule to DNA that carries the same code, and we use it as the messenger from our DNA, which remains intact, you notice, out of the holes in the nucleus, which you can see up there, and into the outer part, the cytoplasm of the cell. And there it gets hooked onto a protein production factory, the ribosome. And with the help of another molecule, transfer RNA, which reads the bases off 3 by 3, because it takes a triplet of bases to code for one amino acid. And you see that what they're going to do now is to join together. So the successive triplets of code lead to the generation of successive amino acids in a chain which is being put together. And amino acids are a more complex set of molecules than nucleotides, and they're able to fold up into elaborate structures when the chains are complete, and then go off to do what they have to do about our bodies, like the keratin that we were seeing earlier. The folding process is inherent in the protein, and this is an extremely important point. The proteins know how to make the structures. Nobody has to tell them to fold up. So these are the levels. This is how the hardware begins to develop from the software. The software leads to molecules, and this is a computer animation which is simulating for us the folding of quite a small protein molecule called pancreatic trypsin inhibitor. It's actually an important molecule because it stops your insides digesting themselves when you're having a meal, which is a useful thing for carrying on. But you can see it wriggling around, and what the program is doing is to have it constantly is exploring contacts with itself and coming together. Now, coming back to this self-assembly thing, the proteins themselves put themselves together. And this is a general theme throughout life. Let's see what this block of this pile of bricks is going to do. That's something like that life does. The bricks know where to go. But there's two problems. First of all, how they know where to go, and the second thing is how they do it. I mean, if you've got some bricks lying here, you know very well that if you come back tomorrow, they'll still be there, unless a builder has taken them and made them into something. But proteins aren't like that. Proteins are very, very small, and they are constantly bashed around. You see, at the protein level, things are so small that you constantly get buffeted by water molecules. That's like this all the time. It's a crazy world down there among the molecules. And we can see that even at a level of the light microscope, if we have a look at what's under here. This is a magnified picture of milk. And these little droplets of fat in the milk, you can see, are moving around the whole time. That's not happening because they are alive or because they're motile. It's happening because they're being bashed by water molecules. Now, proteins are much smaller than that. You can't see proteins under a microscope, not individual molecules. And so they get really bashed from one end of the cell to the other all the time. So that's one part of it. That provides the sort of energy to do the whole thing. But what about the mechanism? Well, we've got some people come down and show you how it works. So our volunteers here are equipped for polymerization. This is a little bit different from the DNA. The DNA was picking up things and sort of copying itself. These molecules are all the same. These are protein molecules, are all the same. You can see they're identically equipped, and they're going to put blindfolds on, and then they're going to start bashing around. Bash around with your blindfolds and see what happens. I'm going to be a water molecule who bangs around if necessary to see what's going on. They seem to have got themselves in a huddle. I'm going to unstick this one and start it off. Whoop. We had a sort of, there we are, they're coming together. You see 4 have already polymerized, that's right. You see he's trying to polymerize backwards, but he can't. There we are. Well done. Have a good look at them. They're all facing the same way. Any idea why? What's sticking them together? Huh? Velcro, yes. And is it one sort of Velcro or two? It's plus and minus, right? And you can guess how it's been fixed. But you see, that's really just how these protein molecules are. They have the right stickinesses, just like plus and minus Velcro, to put themselves together the way they're supposed to be. That's how, in a very simple way, molecules have these instructions to assemble themselves. And you can imagine making a long, long thing, like, for example, the microtubules, which are an essential part of our cell, which is what those little things hanging up there represent, and they provide the scaffolding for the cell, and they're made by long polymerizations of a molecule called tubulin. Thank you very much. You can lift your blindfolds and get back to your seats. And so we've thought about proteins folding themselves up. We thought about polymerizing the proteins, and they've got to build themselves now into the cell. The world of the cell, which this room, in a little way, represents. We can see the microtubules, we've seen the nucleus. This is part of the cell membrane. Here are the lipid molecules, keeping the outside out and the inside in. And these are the channels and receptors and so forth that allow communication with the outside. So if you imagine this room as a cell, but it's more than that, it's filled with membranes. Where you're sitting, it's filled with structures, all sorts of organelles. They've all got to be put together just to make one of the 100 million cells in the human body. So that's just the beginning. Stick out your tongues. Stick out your tongues, please, all of you. That is what a section to your tongue looks like, a wall of cells. I was going to say that it looks pretty in your tongues, but I must say I see some very beautiful tongues, so I won't say that. I think your tongues are really nice. Thank you. And so now, in a moment, we'll move on to the next level of complexity, the building up of cells.
You were working as a waitress in a cocktail bar when I met you. I picked you out, I shook you up, and turned you around, turned you into someone new. Now, 5 years later on, you've got the world at your feet. Success has been so easy for you. But don't forget it's me that put you where you are now, and I can put you back down to me. Don't you want me, baby? Don't you want me? Whoa, 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 whoa.
Benylin. Nothing is more effective without prescription.
Don't you want a fantastic deal on a Fiat Punto?
Don't you want me, baby?
Of course you do.
Whoa, 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 whoa.
Call 0800 717 7000.
Most of the cells, most of the organisms actually on Earth today are single cells. Most of them are pretty small. They're bacteria, like the ones that formed the very earliest sorts of life and are still all around us today. It's an interesting thought that there are far more bacterial cells in and around your body than there are cells of your own. Ever thought of that? But others are very big. This ostrich egg, for example, is a single cell before it starts to develop. But in general, to make larger organisms, you have to use lots of little cells. That's the way they work. And this picture here shows us the beginning of a human being, the fertilization of an egg by a sperm. That little cell, the fertilized egg, just a pinprick, is going to grow and divide and grow and divide. And we'll see a graphic of that, or rather a film of that, in a moment with another organism which develops into not so many cells as us and is the nematode worm. The fertilization happens, and you see the two pronuclei coming together. That's the male and the female nuclei. You can see the separation, and you see the first cell division. And it's going to go on and on dividing, and we're going to watch that happen. Now, as it does it, the same DNA gets packaged into all the cells. So how come it's able to put together an organism? How come we're able to be an organism when we're made of so many cells, all of which contain the same DNA? It's important that somehow the cells know what to do. The way to get at these questions is not to study people in the first instance, because we're too big, we're too complex, and also rather precious. You don't go cutting out bits of people and messing about with them in the lab, but you can do it with simpler animals like this nematode, which is why we see its development here, and like the fruit fly, which has given us a tremendous opportunity to discover these control genes. Now under here we have a fly which is Antennapedia. Can you see what's wrong with this fly? It's a little awkward angle. You can see that this is a leg growing out of its head, which is why it's called Antennapedia. There was another fly which was discovered nearly 100 years ago, which is this one, which has 4 wings instead of only 2. And this was what gave the clue to the idea that there would be control genes that somehow specify the different parts of the body, which of course is exactly what you want if you're going to make this great menagerie of cells into a defined, differentiated structure. And to see a bit about how these genes work, we've got a model here. Looks like I could get somebody to come and help me with this. Would you like to come? Okay. Come and help me put this fly together. You can see there's a fly there, and you can see we've got coloured bits of its body. And the puzzle is to put the bits together. Can you have a go? See what you're doing. Yes, there's the head. And you can see that the various parts are coloured. Now, there's a good reason for that. It's because these parts actually come from strips, from segments in the early fly. Here's a big one that goes here, I think, somewhere. Is that right? Yes, there we are. You can see the legs are appearing. In the early fly, these strips, these segments are laid out, and then they get specified to have different fates. And the genes that specify them are called the genes in the Hox cluster, which define the fates from head to tail in these segments. That's it. I think you were there. Oh, no. I didn't. I can't do this one at all. Perhaps we put that one in. There we are. We're getting there now. You're nearly there. Good. Thank you. That's a great help. Well done. Now, the genes that label these segments are laid out in order here according to the colours, and that's not for an accident, because that's just the way they look along the fly's DNA. Quite remarkably, they're laid out in order along the fly's genome in the same way that in the same order that they're laid out in head to tail on the fly's body. And the same genes actually are found quite widely, or at least relatives of them are found quite widely throughout the animal kingdom. But first, I want to tell you a little bit about how we can see. Oh yes, let's look at this picture first up here. This is how the fly begins. You can see the stripes being laid out. Here, the genes that control the early development of the fly are being made to produce an enzyme which allows us to stain in stripes the areas where they're being expressed. And these are the genes that lay out in equal sort of, they lay out the structure. In fact, a way of describing it, of thinking about it, is building a house. If you want to build a house, what happens is that you make a scaffold, sort of area to work in, and that's a bit like the mother's body, which is incubating this growing organism. And then you start to build the bricks, and then you start to make floors, and you start to divide into rooms, and then you specify the rooms for particular purposes. So you see, making an organism is a little bit like doing that. You get the overall structure first, as these genes are doing in the early part of the fly, and then you, as we saw in the model, specify the regions to do particular things. And what happened with the four-winged fly is that a segment which was supposed to produce just little balancing pieces produced whole wings instead. And in the one under the microscope that we had there before, the area of the head that was supposed to produce an antenna produced a little sort of stunted leg. And the reason for that is that the regional specifiers were wrong. Now, what the regional specifiers go on to do is to control lots of other genes. And it's quite hard to figure out what's going on, but we can often do well to link the genes to fluorescent molecules. This is one that comes from the jellyfish, the green fluorescent protein. And what we're seeing here are now nematode worms, which Duncan is organizing under the microscope. And if we pop the light up so that we can see the worms as well, there you can see what we're actually seeing. These are worms whose head, whose mouth part, that's the mouth where it pumps food in, is stained because there's a particular sort of muscle there which has been linked to this fluorescent protein, and when it's expressed, you can see it. So you see, you can go on doing this throughout the body of a developing animal and see where and when genes are expressed by linking them to molecules that you can see. It's a way of labeling things and showing where they act. Now, what do these genes go on to do, all these control genes? Well, they obviously, sometimes they specify making keratin again. If it's an appropriate part of the body to make hair, then they'll specify keratin. So they'll specify actual parts of the body. But it's much more complex than that. Lots of them will specify signals. They'll specify messages that have to go from cell to cell so the cells can communicate with each other as the system grows. That's what makes it stable. That's what allows the whole thing to regulate itself, so you don't just depend on accident, but constantly it brings itself to the right form by very, very complex systems of communication. And sometimes odd things happen. For example, while you develop, a lot of your cells die. That seems a bit daft in a way that you would do that, but it's actually the most economical way. And when you look at this embryo in the uterus, you see how it's got a little sort of webbed foot, like that. These are really the fingers, they've not been separated yet. And so during early development, cells have to die in between your fingers so that you don't have webbed feet. So all of this is programmed, all this elaborate process is played out, and the toolkit is preserved. Those HOX genes, that cluster of control genes that we saw laid out on the fly, are reused, not in precisely the same way, but in quite analogous ways in laying out our body. They become more complicated. It went through a process where we get multiple copies of those, so it's harder to work out. Again, this is why the fly and the worm are good for working things out. They have smaller genomes, fewer genes, they're simpler to deal with. But by looking at what's going on in those, we can then come and look in our own bodies and find what's going on there too. But there's another sort of thing that happens in developing your bodies. You don't just make the thing sort of growing out as a mass and sort of roughly the right shape. You also have to, some cells have to grow out over others. For example, wiggle your toes. Everybody wiggle their toes. What's going on when you wiggle your toes? You're sending signals from your brain down your back, and there are nerves that start in the back here, from the spine, and run all the way down your legs. Those cells don't get there in the first place as the body is growing. They actually grow out along the other cells or over the other cells. And here we see this happening in a dish. We see a group of nerve cells which are still growing. They're from a growing embryo, and they are sending out these processes just as yours will have done as you grew as a baby, growing nerves out down your legs and wherever you needed them. And if you touch your toes, you can feel the sensation of that, and then of course the signal is going back the other way into your back, and again you have these long processes of nerve cells that are carrying that information. So you see, coming back to our house and thinking about that, when your house is largely built, electricians come in and lay out the wiring. That's like your nerve cells. You also have plumbers come in and lay out the plumbing. That's like your blood vessels, and they grow out over the other tissues as well. And so you find that the whole thing is put together. And how's Max doing? Can Max come on camera for a little while again? That would be great. Good, Max. So you see, Max has been assembled in this way. Nobody told him what to do. There weren't electricians or plumbers. There were just control genes that told the DNA when and where to make materials. But the materials put themselves together, as we've seen. The proteins folding right, sticking themselves together right, making cells, the cells sticking together and developing. And so Max assembled himself. And he did it with DNA that's 99.8% the same as that of the DNA of anybody else in this room. We all share nearly all of our genome. We have it in common from the long process of evolution that's produced us, and we use it to make ourselves. The variations are important, and we'll be talking about those next time. But for the moment, Max assembled himself, and a new human being was born.
The American version of a British series next. Drew Carey is your host for a very witty Whose Line Is It Anyway?